Cell Clinical Research Private Limited was established on April 2006 in Mumbai under the Registration of Indian Companies Act 1956. It is a joint venture between Messrs. City Pharma, KS Copenhagen, Denmark, and Lok Beta Pharmaceuticals Private Limited, Mumbai, India. Cell was established for scientific development and practical applications of novel biotechnological methods in human disease treatment using fetal stem cells and tissue extracts. It is also involved in development of new rejuvenating and anti-aging compositions containing fetal tissue extract. The company has its own state-of-the-art facility to provide continuous electricity and water. The air handling unit, comprising of HEPA filters, regulates the temperature and humidity in the laboratory complex and provides a germ-free environment. The research center of Cell is unique in India with its ultra-modern laboratory complex built in compliance with WHO GMP guidelines and is approved by various organizations. Cell is also supported by various scientific collaborations, novel technologies, and patent inventions in the field of regenerative medicines and stem cell biology. The research team of Cell is represented by highly qualified scientific personnel as project leaders with efficient team members. Scientific Director of Cell, Professor Alexander Kukharchuk, has several years of work experience in scientific and clinical research on stem cells in Ukraine. He has 17 patents on his name and more than 250 scientific publications. All work stages of Cell, regulated by standard operational procedure, comply with the requirements of ICMR DBT guidelines for stem cell research and therapy. Staffs of Cell working in clean class C and B premises have special clothing, which is subsequently worn after taking a shower. Delivery of anatomical material from the clinics is carried out in cold boxes by trained personnel. Supplementary documents like donor's informed consent form for anatomical material, analysis report of donor for HIV infection, hepatitis B and C, syphilis, and cytomegalovirus are recorded in the special register. The cold box containing anatomical material is transferred to a non-classified corridor through the air shower. After cleaning the container's external surface with a disinfectant, it is transferred through the pass box to the preliminary anatomical material processing room of Class T. Before taking the abortive material to the manufacturing department, preliminary processing is essential to detect the presence of microbial contaminants on the abortive material due to the handling in the hospital. The abortive material is washed with sterile solutions and the washed tissue fluid is subjected for rapid microbial endotoxin test. If endotoxin is found to be present in the tissue wash, the anatomical material is completely destroyed. If endotoxin is found to be absent, the anatomical material is then transferred to manufacturing laboratory through the pass box. The manufacturing department is a clean class B zone. Stem cells and progenitor cells are isolated here from different organs of the anatomical material under the laminar hood of clean class A zone according to the patented method of Professor Alexander Kukharchuk. Aliquots of cell suspension are transferred through pass box to the cryopreservation room. Here, a special instrument called cryoplanner with an original program of Professor Alexander Kukharchuk is used for gradually decreasing the temperature of cells from room temperature to minus 196 degrees Celsius without harming the cells. Aliquots with the frozen cells are then placed into the cryorepository until investigation of its biosafety and characterization are completed. For using stem cells in therapy, their characterization, screening, detection of various bacterial and viral pathogens, and the presence of endotoxin needs to be carried out as per the guidelines for stem cell research and therapy to safeguard against potential risks in biological therapy. EmproCell has a special biosafety department. Biosafety laboratory consists of the ELISA laboratory, fish analysis laboratory, PCR laboratory, and cell culture laboratory. Aliquots of each batch of cells from cryorepository are handed over to Biosafety Department for cell characterization and detection of various pathogens. The ELISA laboratory of Biosafety Department is engaged in the detection of the presence or absence of endotoxin using chromogenic endpoint assay, and the results of all the experiments are shared with biotechnologists from the manufacturing laboratory. Endotoxin positive samples are discarded and only endotoxin negative samples are further processed by the biotechnicians of the manufacturing laboratory for the separation of fetal organs and tissue extracts and the isolations of stem cells from it. The Cell Culture Laboratory of Biosafety Department is designed to comply with the requirements of Class B zone. This laboratory is well equipped with advanced and most recent instruments and is engaged in cell culture activities such as cell enumeration, colony formation assay, etc. As suggested by the guidelines for stem cell research and therapy, screening of cryopreserved cell suspensions is carried out in the Pathogen Detection Laboratory of Biosafety Department. The nucleic acids are isolated from cryopreserved stem cell suspension and used for detection of pathogens like hepatitis B virus, herpes virus, cytomegalovirus, HIV, and torch infection by using real-time PCR analysis. Besides, sex determination of cells is carried out, which is significant for practical medicine. EmproCell has a separate sterilization lab. It is well equipped with a MilliQ water system, vertical steam sterilizer, hot air ovens, and ice-matic machine. Sterilization lab is responsible for supply of sterile labware, garments, laboratory reagents, and solutions to other departments. The in-house Research and Development Unit supports the research activities for improvement of processes and development of new protocol for cell separation and fetal tissue extracts preparation. The R&D unit has a fully equipped and dedicated lab in addition to a cell culture facility for development of molecular biology-based protocols and projects for the empowerment of technology. Study of stem cells has led to the creation of a novel treatment for disease conditions in patients. Regenerative medicine is a gifted hope in the treatment of dying patients. Method of regenerative medicine constantly excels on the basis of scientific research. EmproCell is open for the scientific and practical collaboration with other laboratories and clinics.